The polymerase chain reaction, or PCR, is a lab technique that produces numerous copies of a specific segment of DNA in a relatively short period and from a very small sample. This technique is used in many applications, including to help identify persons involved in crimes, to identify microorganisms that cause disease, and to understand evolutionary relationships. PCR is a three-step process that is repeated over and over and produces identical copies of a target sequence. In a small tube, DNA containing the target sequence is mixed with deoxyribonucleotides, or DNA bases, adenine, thymine, guanine, and cytosine. A heat-stable DNA polymerase enzyme called Taq polymerase, buffer, and small segments of DNA called DNA primers. These are the components needed to copy DNA. Taq polymerase is an enzyme derived from bacteria that thrive in hot springs and hydrothermal vents. Because these bacteria live in an extremely hot environment, their polymerase enzyme, the enzyme that helps replicate DNA, can withstand the near boiling temperatures necessary to complete a PCR reaction. To perform PCR, you need a device called a thermal cycler. This machine raises and lowers the temperature in pre-programmed steps as required for copying DNA. The PCR components, target sequence, deoxyribonucleotides, Taq polymerase, and DNA primers, are added to a PCR tube. The tube is then placed in the thermal cycler and the PCR process is initiated. The PCR process. 1. Denaturation. A process that causes complementary DNA strands to separate. 2. Annealing. Primers attach on each end of the target sequence. 3. Extension. Taq polymerase adds complementary bases to build the new DNA strands. The PCR process is repeated, usually 30 to 40 times, each of which produces identical copies of the target sequence. To denature the DNA, the thermal cycler heats up, which causes the hydrogen bonds that hold the DNA double helix together to break, creating two single-stranded DNA molecules. The thermal cycler then reduces the temperature enough to allow the primers to attach, or anneal, to each end of the target sequence of DNA. The primers are designed to specifically bookend the target sequence and provide a starting point for Taq polymerase to begin copying the DNA. The thermal cycler then raises the temperature a little bit to enable the extension step. In this step, starting at the primers, Taq polymerase adds complementary bases to build the new DNA strands. The three-step PCR process begins again, and the thermal cycler raises the temperature again to separate the DNA strands. The temperature then lowers to allow primers to attach. The temperature increases, and Taq polymerase synthesizes new complementary DNA strands. This sequence of 3 steps is repeated until enough DNA has been produced for use in analysis. This usually takes about an hour. PCR is an amazing process that has revolutionized the fields of forensics, scientific research, medicine, and more. Scientists continue to find groundbreaking and life-saving applications for this technology. Can you think of ways in which PCR could change the world?